So welcome everybody. Hopefully everybody can hear me. This is Tim, one of the co-hosts for today. Thanks for joining us. This is one of our first off-cycle kind of bonus meetings, if you will. Normally we're here at meeting the second Wednesday of the month, and then which we did this month, and then we're doing this one as a kind of a trial. But glad we got such a good crowd, and obviously it's a great topic, and looking forward to Carolina's return visit from just a year ago. And Yvonne will discuss that in just a moment. A bunch of board members here present today on today's call. Just call you out. If I miss anybody, I apologize, or you can just raise your hand. Tim Murphy here, Cheryl Ebner, Yvonne DeGraw, our programs chair, Mary Howe Grant, Bob Lilly, Joseph Stronks, John Fishpaw, Steve Skopatz. So that's who I see on our roster from the board. And thanks everyone else for being with us today. Thanks for your donations for today's meeting during your registration. It really helps with our ongoing administrative expenses and our donations for our various programs. Please consider making donations to our scholarship fund. That's kind of our mantra here. We look to make a major contribution to the Santa Barbara Scholarship Foundation through the SEC every year. And look forward to your consideration on that. So I think with that, I'll turn it over to Yvonne here to talk about upcoming events and today's event. Go ahead, take it away, Yvonne, please.
Thanks, Tim. I just wanted to, circle back to our January meeting and say that we were happy to have Matt Heverly speak to us about the Perseverance rover, and it landed successfully. So if you haven't already watched the video of the landing, it's pretty amazing to see video of a landing on another planet. So go watch that. And to talk about our upcoming meetings, on March 10th, we're going to have Dr. Ayal Amir. He's the CEO of a startup called AI Incube. He's going to be speaking about smart cities. And so we'll be having the email for that go out shortly. In April, we normally have our science fair meeting. They are having a science fair in Santa Barbara County, but the number of entries is understandably lower than normal. So we haven't yet decided what we're going to do about that April 14th meeting. It's possible we might have that as a bonus meeting toward the end of March, just to recognize the winners of the science fair. But we'll be letting you know by email. So if you're registered for this event, you should be getting those emails. Today, we're happy to present this bonus meeting, February meeting, to provide an update on vaccines and viral variants. Our speaker today is Dr. Carolina Arias. Dr. Arias spoke to the Science and Engineering Council in March 2020, just before the SARS-CoV-2 virus reached Santa Barbara County. It was, I think, one of the last things that a number of us did in public in a group of people. She joined the UCSB faculty in late 2016 and is an assistant professor in UCSB's Department of Molecular, Cellular, and Developmental Biology. She earned a PhD in microbiology at New York University Sackler Institute for Graduate Biomedical Sciences, where she studied virus-host interactions in herpes virus and pox virus. Dr. Arias has worked in the labs of Dr. Don Gainham at UC San Francisco, the Novartis Institutes for Biomedical Research, and the lab of Dr. Joseph DeRizzi at UC San Francisco. And if you have questions during today's talks, you can type them into the Zoom chat area, or you can ask them after the talk when you can unmute them yourself and ask the questions directly. But today, we're pleased to introduce Dr. Carolina Arias.
Thank you, Yvonne, and thank you, everybody. I'm just gonna first say, I prefer to call me Carolina, so you can just call me by my first name. And if you have any questions, just either, as Yvonne said, type them or just let me know at the end. And let me start by sharing my presentation. So just give me 1 second, right here. So can everybody see my slides? Okay, So thank you so much for inviting me again. As Yvonne was just saying, the last time I talked to you guys, it was March 10 or 11 of last year. And I clearly remember that day because, as I'm going to mention to you in a moment, that was the day that SARS-CoV-2 was declared, or that COVID-19 was declared a pandemic. And we'll talk about that in just a moment. So I'm very happy to be here giving you an update, just a short year later, or a very long one for most of us, an update on vaccines and viral variants, which are inundating the news basically every day. So this was my first slide from last year, and I decided that I was going to leave it for this year, because every time that I open the news these days, I just feel this rush of like, oh my God, what's going on? But we have to remember, don't panic. There's a lot of information that is coming out, and I'm going to try to kind of streamline it and dissect it for you today. But the main message of everything that I'm going to tell you today is don't panic. And the same message I gave you last year, I give you again this year, that I love these words by Marie Curie, that nothing in life is to be feared, it's only to be understood. And now is the time to understand more so that we may fear less. And again, in all of these presentations, my main goal is to transmit the information in a very direct way, trying to help you see also what us as scientists understand about this disease and understand about these vaccines, so that we can calm down and reduce some of the fear that a lot of the information, misinformation and disinformation can cause us on a daily basis. Okay, so today I'm going to give you 3 main topics. The first one is we're going to talk about a COVID-19 update, although I know all of us are glued to these numbers. I'm going to tell you what's going on in our county and in the world. We're going to talk about vaccines, and here is where I'm going to spend most of my time. Kind of giving you a primer of what vaccines are, of what the available vaccines are for COVID-19, what is in development, and what does it mean, a lot of the terms that they're throwing at us these days. And of course, we're going to talk about some of the viral variants which have arisen in the past months and are causing some concern in the community. And many of you may have read the LA Times or the New York Times or any newspaper, and you probably saw some information coming out in the past few days about California. Okay, so let's start with the COVID-19 pandemic. So as I told you, these was the numbers that I showed you a year ago. So we had still only 400 cases in the US. We had none in Santa Barbara, so we didn't report any cases until later in March. And this date, March 11, was when the World Health Organization, the WHO, declared the COVID-19 a pandemic. So basically this was when all the protocols to control the pandemic started. Unfortunately, the situation, as we all know, is dire. From 400 cases in the US, we are now, I took this information yesterday from the Johns Hopkins website. And we are at over 28 million cases and over 500,000 deaths, which puts us in the first place for cases and deaths in the world. We have globally over 2.4 million deaths and over 100, I'm pretty sure by today it's probably 112 million cases. So again, we are not close yet to control this pandemic, and it's causing unprecedented suffering all around the world. In the US, I don't need to tell you, but again, the situation is pretty bad, and it changes with, of course, the area where you're at. Again, unfortunately for us, LA is one of the counties that has the most cases and the most deaths caused by COVID-19. The rates of why the disease is so severe in certain counties versus others, it depends on what the population is, what is their access to testing and to treatment, what is the density of people in the urban or rural areas. There's a lot of factors that are going to affect how a disease like this is transmitted, of course. So let's look at the Santa Barbara County. Overall, our county has been not too bad, let's say, in terms of numbers, but it's still causing, this disease is causing suffering throughout every single community in our county. We have over 31,000 cases confirmed, and 396, which by today are probably already 400 deaths. And we have received 78,000 and change vaccines. So again, the numbers are improving in our county. We are seeing the cases and the deaths drop from the second surge. I'm not showing you here the peaks that we've seen, but we know that we just finished a second surge. The numbers are low, things are coming down. But as I'm going to mention also, it is important that we are still vigilant and that we're still careful to prevent the recurrence of a third surge. So we cannot drop our guard right now. It is imperative that we continue taking care and continue the measures for prevention, the washing hands, wearing a mask, avoiding crowds, etc. And of course, receiving vaccines if possible. So that brings me to what are we doing as a country and in general in the world to confront this pandemic. So usually to deal with this kind of diseases and with this kind of pandemics, you can have basically three avenues that you can take. One is you can diagnose it. So if you remember last year when I talked to you, we had no tests for COVID. It's like even if you had, cough and fever and shortness of breath, it was very difficult for you to be able to access a test. You basically had to be in the hospital to be able to be tested. Fortunately, the situation changed, and now we have testing that is free for some members of for most members of our community in different places. We have a program here at UCSB that allows us to test our faculty, staff, and students that are currently on campus, that are not many. We have really, the country has tried to have widespread testing. It is still challenging in some places, but it's definitely a million miles away than what we were last year this time. So yes, diagnostics help. We are not going to be able to end our diagnostic programs anytime soon. We have to continue monitoring our community, and this is something that we still have to work on making it accessible, free if possible, and having it widespread. Another avenue is treatment, and we've also had a lot of development on this the last year. Fortunately, there were a lot of companies and institutes and research groups that have focused on developing some sort of treatment. If you see, although the cases of COVID-19 have increased dramatically in the past months, the deaths have not. The deaths have actually stayed low comparatively to the number of cases that we have reported. That is because we have more diagnostics, so we can actually capture more cases, including those that are in asymptomatic individuals. And it's also because we now have better options for treatment. Doctors know how to manage these patients better. They have some options to offer them some help, especially in severe cases. We have heard about antibodies and drugs that are now approved and available for treatment. I'm not going to spend more time on this particular aspect, but the good thing is that there is hope for treatment. They continue being developed, and there are a lot of groups actively working on this avenue that is going to help us mitigate the pandemic. But of course, one thing that we need desperately, and that fortunately we have options, is for prevention. So the non-pharmaceutical intervention measures, the wearing the mask, washing your hands, the lockdowns that we have, the social distancing, avoiding crowds, etc., has helped dramatically control this disease. It has mitigated this pandemic at some level. And right now, this kind of sharp decrease that we had in the second surge that we just went through cannot be completely attributed to the fact of having vaccines. It's actually most likely that this controlling the cases, the controlling the transmission, is due to the behavior of people, right? There is a change in the approach, and there is a change probably in the mentality, because the vaccines are right next to us. The vaccines are coming, they're right here. It would be a shame to get COVID right now when the vaccine is available. So that probably helped people become more conscious about these non-pharmaceutical interventions, and be also more aware of wearing a mask and washing your hands, etc. Of course, we understand more transmission, so we can prevent it better. But what is key for us to be able to deal with COVID-19 is having a vaccine. And as I said, fortunately, we have some options. So before I go into what is going on for COVID-19, I wanted to take some minutes to talk about what are vaccines. What are we talking about when we say these vaccines that we're hearing so much about? Well, every time that your body is exposed to a pathogen, a virus, a bacteria, or a parasite, it elicits or it mounts an immune response. This immune response is going to help you deal with that pathogen, try to eliminate it, and it's also going to offer you some protection for future infections. So something that is very important is what is illustrated here. So here where I'm showing you four different pathogens. And you see that each one of those has different patterns. Those patterns are the ones that are going to be recognized by your immune system. And your immune response is going to be mounted and it's going to be elicited accordingly to those particular, let's say here, shapes that these pathogens have, these particular motifs and these particular patterns. So all of that is to say, if you have an infection, in this case, by measles, you're going to develop an immune response and you're going to generate antibodies that are specific for that measles virus. They're going to be able to neutralize and eliminate that pathogen and offer you protection for this particular measles virus in the future. However, if you are now exposed to flu or to chickenpox, the patterns on those viruses are very different, and you're going to need to mount a new immune response that is going to protect you against those pathogens. That is a long way to say, if you're exposed to a new pathogen, in this COVID-19, having previous infections or previous vaccines for influenza or for chickenpox or for measles is really not going to offer you a protection against this new pathogen. You have to mount a new immune response that is going to deal with this particular pathogen and that is going to be specific for this particular virus. And that is what we're going to take advantage of to generate a vaccine. And that's what's happening right now with COVID-19. So how do we take advantage of that? Well, we know that the response is pretty specific. Basically, you are exposed to a new pathogen, you're going to generate antibodies for that particular pathogen. So we can kind of train your immune system to think that you were exposed to that pathogen by offering it little pieces of that particular pathogen. In this case, little pieces of the COVID-19 virus, the SARS-CoV-2 virus. As I'm going to tell you, we have other approaches that involve the entire virus, but for this example, we're going to say just this little green piece of this pathogen. So your body is going to see this as foreign. It's going to recognize that there's something in your body that should not be there, and it's going to mount a specific immune response against this pattern here that is recognizing. These antibodies are then going to offer you protection for this virus whenever you are exposed to the real pathogen. So it's again just a training exercise for your immune system that is going to help you respond in the case that you encounter, in this case, the SARS-CoV-2 virus. So what types of vaccines are we using? So I just told you that we use different pieces of the virus to train your immune system, but what does that look like? What kind of pieces do we use, and how do we know what to use? I can tell you that this comes from years and decades of research in vaccine development. And we know that there are some strategies and some alternatives that we have to develop a vaccine that is going to successfully elicit an immune response and provide you protection for that particular pathogen. So some of the strategies that we're using for SARS-CoV-2. So this is the SARS-CoV-2 virus that all of us are very familiar with right now. We see it basically every single day in the news. Material is instructions that the virus has to make more viruses. This genetic material is protected by different proteins that form a shell around this genetic material, this RNA, to protect it and just preserve it. One of those particular proteins is something that we have heard about a lot, which is the spike protein, these projection-like proteins that we find in the SARS-CoV-2 virus. These spike proteins mediate the recognition of the receptors in the cell. Basically, it's a kind of key and lock mechanism. The spike is going to be the key. The receptor in your cells is the lock. Once the lock is open, the virus can go inside of the cell and infect it. And when it infects, as I'm going to show you, it just hijacks the entire machinery of the cell and it converts the cell into a viral factory. So we know that a lot of the immune responses, or we know that the spike protein can elicit a very robust immune response. So there's a lot of focus on using this particular piece of the virus, this spike protein, as the little piece that we're going to use to train your immune system to recognize this virus. So that's one of the alternatives that we have. And there are some vaccines that are right now in development that use a protein subunit, a little part of the spike protein that we're going to use to train your immune system. We can also use the instructions that are in the genetic material of the virus that are going to basically tell us how to make this protein. We can use just the genetic information to make the spike protein, and we can give that as an mRNA vaccine, and I'm going to give you more information about this, or we can deliver it using another vector, these virus vector vaccines, and I'm going to tell you a lot more about these two strategies. I'm not going to talk much about that right now. But this is just basically giving the instructions to your cells so that your own cells make the spike protein. This is not going to cause any disease, and it's going to, again, train your immune system to recognize the spike protein whenever it sees it. We can also use virus-like particles, and in this case, instead of using just a little piece of the protein, just a little piece of the spike, we use something that looks like a virus, that is decorated on the outside with the proteins that a virus would have, but that it doesn't have any genetic material, so it cannot replicate in your body. It cannot make more viral particles because it lacks the instructions to make viral particles, but it's still going to train your immune system to respond. We can use inactivated virus, which is basically we just kill the virus and we're just going to give you dead virus, and that is going to elicit some immune response without the virus replicating or causing disease. And we have some vaccines that have been approved in the past for other viruses that use this mechanism, as well as some of those that are live attenuated. And what this means is that the virus is still alive. It can still replicate, but it doesn't cause a disease as severe, it doesn't cause a disease as the wild type or as the normal virus would. So we just basically don't kill it, allow it to replicate, but it is not going to cause disease. And again, we have some vaccines that use this strategy that have been approved for other viruses. Each one of these vaccines, or each one of these strategies, is being used right now for development of a vaccine for COVID-19. But I'm going to tell you which ones we have, and as I said, I'm going to focus on two of those strategies that are the ones that are approved or close to be approved. Okay. So one of the questions that I get the most is, but what happens? How do you develop these vaccines? And we talk about vaccine development is that it takes years, if not a decade, to develop a successful vaccine. There are a lot of steps and a lot of care that goes into developing a vaccine. And these are the phases that you can that you have when you are making a new vaccine. You have to explore how to develop it. So again, we have to go and choose what are we gonna train your immune system with. Should we do a live attenuated? Should we do a protein based? How do we start? So this exploration phase takes a long time until you find something successful. Once you find something that is successful, you have to evaluate it in the clinic. So you do preclinical assays where you evaluate the safety of your vaccine, especially in animal models, and you see if there are any adverse effects that are being elicited by just giving this vaccine. Once that is clear, you go into clinical development, and that means you go and evaluate the efficacy and the safety of a vaccine in humans. Once you pass all these hurdles and the vaccines are effective and safe, you have to go into regulation regulatory review and approval. And this means the process that we are becoming more familiar with, which is the evaluation by the FDA. The FDA and other agencies are going to say, okay, is the vaccine causing adverse effect? Is it as effective as they're claiming? What is the evidence? Where is the data? Show me the data that supports your claims. Once that is approved, you go into manufacturing. So companies are not going to waste millions of dollars making vaccines that are not going to be approved. So you wait for the approval process and start manufacturing. And of course, you go in through a very rigorous quality control system. So I just told you a lot of steps that are very time consuming. So how come we were able to have a vaccine for a virus that we just discovered last 2019? It's been about a year and we have vaccines that are approved. What happened? Did somebody take shortcuts? Did we skip any of these steps? And the answers are no. There were no shortcuts. There were no steps that were skipped. There were a lot of collaborations and a lot of scientists that came together to help speed up this process. So for the exploratory part, we have had decades again of research on vaccine development. And there was actually some research from the past 10 and 20 years for development of vaccines for other coronaviruses. So people already had some ideas on what should we be looking for and what the strategies could have potential for the development of these vaccines for SARS-CoV-2. Remember, this is not the first coronavirus that we encounter as humans. We've had other pandemics and other epidemics caused by coronaviruses. So once people had some strategies and some, again, a lot of research that has happened and a lot of advancements that have happened in the past decade, all came together and all coincided in helping us speed up this exploratory process as much as possible. I can tell you that the preclinical and clinical development are the lengthiest parts of this process. As I'm going to tell you, 2 of the vaccines that are approved now, it started their clinical trials in July. So these 2 steps, the preclinical and clinical development, were done as rigorously as they were done for vaccines that take years to develop. So there were no shortcuts here. There was no data that was skipped. There was no step that was missed. The process proceeded the same way as it has with other vaccines. Another step that has been speed up is the regulation step. These entities, the FDA and other regulatory entities, have really, again, like buckle down and come together quickly and review all the data and prioritize all of this data that needs to be processed and reviewed carefully. So just because they're doing it quickly doesn't mean that they're being sloppy. They're actually looking at the data and considering really seriously the benefits of these vaccines. And the other 2 steps that kind of were shortened were the manufacturing and quality control. So right now, companies are not waiting to have this regulatory approval to start manufacturing. So there's a lot of investment right now that these companies had to be able to make as many vaccines as possible in the shortest amount of time to be able to accommodate the supply that we're going to need globally. And of course, the quality control is already included in this manufacturing steps. So no steps were skipped. I think we just learned how to do it more efficiently, and people collaborated, and people are all together in this process to try to get a development of a vaccine as quickly as possible. So what are these clinical trials that we hear so much about? So we have heard about vaccines in phase 1 trials, vaccines in phase 2, phase 3, etc. So what that just means is the moment or the size of the study that is being made and the questions that are being asked for different stages in the development. In phase 1, you're going to start looking at the adverse effects in humans. So if we give this vaccine to somebody, do we start seeing that they have any manifestations that we were not expecting, or are they responding okay and as expected? In phase 2 and 3, we start seeing more about the efficacy. Do we see the responses that we expect in people? Again, keeping a very close eye on any possible adverse effects. Do we see that the vaccine is causing the response that we expect, that are people generating the antibodies, etc. In phase 3, we are comparing, of course, those, as we know, those groups that receive the vaccine with those that receive a placebo. And I'm going to give you some examples of that in a moment. And we see if the vaccine is really preventing the disease. Do we see lower cases of the disease that we're vaccinating for in the group that received the vaccine versus the group that did not? And of course, once we pass all of these ranges, the vaccines can be approved for broader use. So once we see that the vaccines are safe, that they are effective against the disease, they're not going to cause any harm, and that the benefit is higher than any of the risks, it can be approved for broader use. So the other term that we are hearing a lot is this vaccine effectiveness. So we hear that some vaccines are 50% effective, some are 60, some are 95, some are 75. What does that mean? So vaccine effectiveness is literally the proportional reduction in disease among the vaccinated group. What do we know for COVID-19? How was the efficacy of the vaccines for COVID-19 evaluated? Well, it was determined as a reduction of mild to moderate symptoms. And we're going to see what that means in just a moment. Another kind of confusion when we're talking about these numbers is often thought that a 95% effective vaccine means that the vaccine is effective in 95% of the people. And that is not the case. So I'm giving you the example here. What is a vaccine that is 90% effective? What that means is that the vaccinated group experienced 90% fewer cases than the unvaccinated group. So it's not that it worked in 90% of the people, it's that it prevented 90% of the cases that we got in the unvaccinated group. Okay, so just as a perspective, the vaccines that we have for COVID-19 right now are performing better than we expected. They're performing absolutely better than many other vaccines that we have out there. A clear example is the flu vaccine. There are years where the flu vaccine is somewhere between 40 to 60% effective, but it's still, even with those numbers, it prevents serious cases of the disease and it offers protection for the entire community. So how does this look for one of the vaccines that is approved right now, which is the Moderna vaccine, and it's very likely, if some of you have already received it, it's very likely that it's one of the Moderna vaccines. And we're going to talk about how this Moderna vaccine works in just a second. So this is a clinical trial for the Moderna vaccine. So 14,550 people received the vaccine and 14,598 received the placebo, which means it doesn't do anything. Then they looked at symptomatic COVID, and by that it means symptoms. Anything that could be related with COVID-19, which we know is a long list of symptoms, anything from fever, sore throat, a stomach problem. There is a lot of symptoms that could be associated with COVID-19. So in the one group that received the vaccine, there were 11. When you compare that to the placebo, that is 185, there is significant protection for this disease in the group that received the vaccine. I think one of the most important things, and what I really want to highlight here, is that in the placebo group, we had 30 individuals with severe COVID-19. These are people that require hospitalization, ICU, and again, were severe. There were no reports of severe disease in the group that received the vaccine. And that is really important. That is what we want to prevent. We want to prevent hospitalizations. We want to prevent serious outcomes of this disease. So the vaccine is doing its job. We can all probably deal with some of the mild symptoms or moderate symptoms that they're reporting, but we really want to protect our population from hospitalization and long-term damage. So the other question that I've been getting lately a lot is a COVID vaccine 70% effect even worth taking? And the answer is absolutely yes. It is absolutely important to get the vaccine. I'm just going to give you an example, and we're going to talk more again about this vaccine in a moment, which is the Johnson & Johnson vaccine. It is reported to be 58 to 72% effective depending on the population. And that is something that it's important to consider. So how are we going to compare the Johnson & Johnson vaccine and the Moderna? We're saying that the Johnson & Johnson is like 70% and the Moderna is 90%. Well, the studies for these two vaccines were done at different times and in different populations. These numbers, the 58 to 72% for the Johnson & Johnson, are from 3 different areas in the world. The lower, the 58%, was in South Africa. And now we know that there are some strains out there that seem to be less sensitive, and we're going to talk about that at the end. Doesn't mean that they are not working, but they just seem to be less sensitive to these vaccines. So by the time that the Moderna's vaccine was being evaluated, these strains were not circulating. And again, they were done in different regions of the world. So there are a lot of different aspects that are going to influence the effectiveness of this vaccine, the population, again, the strains that are circulating, the reports, how accurate are the reports, etc. However, this Johnson & Johnson vaccine is 85% effective for severe COVID-19, which is again what we really want to protect for. And even more important is that there were no severe cases reported 49 days after vaccination. So there is protection by these vaccines. There is the reduction, again, in what we want to reduce, which are the severe cases. So although the numbers don't sound, or sound too different, 70 and 95, is still a vaccine that is effective, is still a vaccine that is offering protection. And as we're going to see, it is a vaccine that is going to provide some options for some regions in the world that cannot access certain types of vaccines. So it is still, a great vaccine, and there are some avenues that people are trying to take to increase the effectiveness a little bit. So what is in the pipeline? So what's happening with these, the development for SARS-CoV-2 or for COVID-19 vaccines? It's been a race. I think probably the most labs and the most institutes all together at one time trying to solve one problem for humanity. So we have a lot of different vaccines that are at different stages of this process that I just mentioned. Some are in phase 1, phase 2, or phase 3 of their clinical trials. Some are authorized, as we're going to talk about in just a second, for early or limited use. Some are approved, and there have been some that are abandoned. Why are vaccines abandoned? Well, maybe the strategy that this company or this institute used did not elicit the level of protection that they were expecting. Or as in other cases, maybe the resources that a company had were limited and decided to reroute to treatment rather than prevention. There are a lot of different reasons why these vaccine trials can be abandoned. So it's not just because they found some adverse effects, but there are also all sorts of conditions that may be fulfilled to continue the trials. So what's out there? So this is a list. Oh, and I forgot to mention that I really recommend, if you have time and you're more curious, to go to the New York Times, and they have this wonderful coronavirus vaccine tracker. They have a lot of information about vaccines. They have a lot of information in the development and the process and where is everybody at. So this is a list of different vaccines that are going on right now. So as I told you, there are a lot of different strategies that are being used. Some of them are delivering mRNA. Some of these are delivering viral vectors with a little piece of the viral genome in them. We have some that are using just a little piece of the protein, of the spike protein, and we have some that are using that inactivated virus I also mentioned. Most of these that I have in the list here are in phase 3, which means they are evaluating the effectiveness in the vaccinated versus the placebo group. And some of them have already been approved in some areas of the world or considered for emergency use or limited use in some other countries, including the US. So we're going to talk about now there's a topic that is going to help us understand a little bit better how these vaccines that I'm going to mention, the mRNA and the vector base, work. And so I want to take a little bit of a detour here, and we're going to talk about biology. So I'm going to give you a very short biology class. And so this is what we call the central dogma. right? So how we go from DNA, RNA, and proteins. So I'm sure that all of you know that your genetic material, the instructions that make you you, are encoded in your DNA. The DNA is this blue squiggle right here that is in every single one of your cells inside of a compartment that is known as the nucleus. Again, the instructions for all the proteins that you need for function and that your cell need for function are encoded in your DNA. But unfortunately, they cannot be read by the machinery that makes the proteins, which are the ribosomes. The ribosomes are in a different compartment in your cell called the cytosol. And so for the DNA to be able to transmit that information to the ribosome so that it can make proteins, it needs what we call a messenger RNA. This messenger RNA is essentially a copy of your DNA that is going to contain the instructions that are written now in a language that the ribosome can understand. And this mRNA is going to leave the nucleus and is going to reach the ribosome in the cytoplasm, while your DNA is going to stay here protected in the nucleus. Something that is important to know at this point is that viruses use this process that I just told you, this transcription from the DNA into the messenger RNA and this translation from the messenger RNA to a protein. They use it for their advantage. They don't have the machinery, particularly ribosomes, to carry out this process. And so when they infect a cell, they make their own messenger RNAs, their own mRNAs, and they translate it using your ribosomes. That's why you often hear me say that during infection, a virus transformed the cell into a viral factory. So we have taken advantage of that fact to develop, or researchers have taken advantage of that fact to develop some of the vaccines and have used this as a strategy to develop successful vaccines for COVID-19 and for other viruses. So let's talk about first some of the two of the strategies that I mentioned about for the development of SARS-CoV-2 vaccines. And the first one that I want to tell you about is these messenger RNA vaccines. It's probably the first time that you hear about them, but I can tell you that they're not new. They've been in development for a long time. And there's been a lot of studies about these mRNAs and how to use these mRNAs as a vaccine. So here what we do is, if you remember, I told you that this is the SARS-CoV-2 particle, and this squiggle right here is the genetic material of the virus. So what we're doing is taking a piece of this genetic material, of this RNA, and now we use it to make one of those messenger RNAs that are recognized by the ribosomes. And we can deliver these by protecting it in a lipid shell. So basically, we're just giving you a little piece of genetic information of the virus that is not going to cause disease, that is not going to generate any viral particles. It's just a tiny little piece of the instructions that are required to make the spike protein. Again, this is spike protein, or let's see how that works. So we have the vaccine particles right here, and we can deliver them. When we deliver them, they fuse with your cell, and they deliver that genetic material into the cytoplasm of your cell. This genetic material never reaches the nucleus. So one concern that some people have expressed to me is like, is this genetic material going to interact with mine? And the answer is no. They are physically separated in two different compartments. And this mRNA is not going to access the cell nucleus and is not going to alter your genetic information. That little piece of RNA is going to be used by the ribosomes, or it's going to be translated by the ribosomes to make the proteins. These proteins are going to be the spike proteins from the virus, and now your cell is going to display them in the surface. That doesn't do anything to your cell. Your cell is just sending a protein up there that it found that it was made from the ribosomes, and it was found an mRNA in there, and it just made it. The same way that a virus would use the machinery to make its own proteins, but again, without the virus in there. These proteins are then going to be displayed. And if you remember, I told you that some of those patterns are recognized by the immune system. So the immune system is going to generate antibodies that are going to recognize this spike protein. And that is going to train your immune system so that the next time that it finds this protein in the context of a virus, it's going to be able to neutralize it and eliminate it eventually. So that's This protein here, the one that was made by your cell and expressed by your cell from this little piece of RNA, is the trainer of your immune system and is what ultimately is going to offer you protection. These RNAs are pretty labile. They are very sensitive to the environment and they degrade very quickly. And eventually, this mRNA is going to be turned over and it's going to be eliminated from your cell and you're going to stop making the protein. Your cells are going to stop making the protein and that is it. But the antibodies that were generated against that spike protein will remain in you. There are some cells that are going to be trained to recognize those spike proteins so that when you find the virus, you can be protected. So what do we know about the vaccines that use this mechanism? And again, this type of vaccine has been studied for a while. And one of the main problems that we had for their use was the delivery. So finding what lipids to use and finding this shell that was going to protect it from the environment was pretty difficult and was one of the steps that took the longest in the lifetime of these mRNA vaccines. So we have now 2 of these that have been approved, one from Pfizer and one from Moderna. Both of them are FDA approved for limited use here in the US. The Pfizer can be stored only in the freezer, and again, is because of the sensitivity that this mRNA has and how labile it is. This one requires 2 doses, and the clinical trials for this vaccine started in July of last year. It is known to be 95% effective, and right now they are completing their trials in pregnant women, which is some information that so far we do not have for the efficacy in this group and the safety in this group. The Moderna vaccine is using the same concept, this RNA that's going to be delivered in this lipid shell. It requires refrigeration and freezer storage, so it's a little different and a little friendlier for delivery and storage than the Pfizer. And all of that depends on the composition of the shell that they used to pack that RNA. We know that it's 94.5% effective, and right now they are, I think, finishing their enrollment of trials in adolescents, which is another group that we have no information. Children and adolescents, we don't know how well these vaccines work and how safe they are in these groups. So what are some of the adverse responses? Well, we know that there are some mild responses after injection. People report, the redness and the pain in the arm. For the second shot, there's been more reports of like headaches and lethargy and people just feeling kind of overall sick. But this goes away after a day or a couple of days. It's not something that's going to be a prolonged display of symptoms. People after that report feeling fine and don't report major effects or major adverse effects. We know that there are some reports of anaphylactic shock, particularly in people that have reported previous events of severe allergies to other medications or to other vaccines. And this is less or around 10 per million people. And again, they are related to prior events of severe effects. So I think the first thing that I did this morning when I woke up and saw the news was kind of, yay! Many of you saw this news, this piece of news today that the Johnson & Johnson vaccine has has met the requirements for FDA approval or FDA authorization. All that means is that the FDA was reviewing the data since earlier this month, and they finally saw that the vaccine or that the data that supports this vaccine effective is right. It is the type of data that we should be seeing, that the efficacy that they report is correlates with data that they're showing, and that the safety of it as well. So it is pretty exciting because it offers a much needed option right now. The supplies of the Pfizer and Moderna vaccine, as we all know, are kind of dry, and it's pretty hard to get a shot, and we need something that is going to help us vaccinate our communities. So this is a great step in the right direction. So what are these vaccines? So these are a little different from the ones that I just told you from the mRNA vaccines. In the mRNA vaccines, we're delivering just that tiny little piece of the genetic material, that tiny little piece of the RNA in a lipid shell. In this case, we are taking the same region. This is also for the spike protein. But now we make it into DNA and we protect it or deliver it using a virus. This is an adenovirus that usually would cause a common cold. And in this case, we replace the genetic material with this little piece of the SARS-CoV-2 genetic material. You can think about this strategy as a Trojan horse, right? So the cells, as I'm going to show you, are going to take the virus. The genetic material is going to be delivered, but is not going to cause any disease at all. This has been previously used and approved for vaccination for Ebola, which was, again, a great advance for the mitigation of this disease in marginalized communities. So what happens? So the virus comes, it interacts with the cell, is taken by the cell, and it delivers, again, that little piece of the genetic material, which ultimately is what is going to result in the generation of the same mRNA that I mentioned before. So this is just another way to deliver this messenger RNA that is ultimately going to result in the expression of the spike protein, the display, and the training of your immune system to recognize that. Some of you may ask, why so much emphasis in the spike protein? Why are we so interested in just, training our immune system for that? Well, it is maybe likely that this protein is the first thing that your immune system is going to see when it encounters the pathogen. It is displayed in the outside of the virus, and your immune system is going to see it. And not only that, but the antibodies that bind the spike protein can neutralize the virus, can prevent it from actually recognizing the receptor in the cell and prevent it from infecting. So it's a very good strategy to train the immune system and to have a vaccine that is going to be effective. And we're going to talk about that in a moment, of why is that important. So what do we know about these virus vector vaccines? Well, as I said, the main one that there's actually two that are pretty successful and encouraging. One of those is the Johnson & Johnson. The FDA is going to provide a recommendation on Friday, and what that means is what is the level of approval and how do they recommend to use it in this emergency situation. One great thing about these virus vectors is vaccines is that they are more rugged than the mRNA. The mRNA, as I told you, is pretty sensitive, right, to temperature changes. You have to have a cold chain. I don't know if you guys heard about hundreds of vaccines that had to be tossed because the cold chain was not successfully maintained, and they were exposed to temperatures that were not optimal, and they were just discarded because they were not working, or we could not afford investigating if they were still working or not. The Johnson & Johnson, again, is stable in the fridge for 3 months. And that is absolutely great news for communities that don't have the capacity of shipping or storage at super ultra low temperatures. So this is going to be optimal for communities like, for example, in Latin America or in Africa, where you have to reach some rural or remote areas where you cannot have this cold chain be maintained all the time. So this is absolutely, you have no idea how happy it makes me. I'm from Colombia originally, and I was always worried how they were going to do it there. And now it's like, this offers a viable alternative that can help deliver it to these areas. The other great thing is that it's one dose. So one of the major caveats that we have with 2 doses is that a lot of people are not compliant. They take 1 dose, but they never return for the second one, which is pretty bad. You don't get the same level of protection if you skip 1 dose. So having 1 dose is pretty good. Right now, they're testing 2 doses to see if they can boost the effectiveness a little bit. But as I mentioned, a 72% effective vaccine is pretty good. It is not unheard of. It's actually the effectiveness that we have for some vaccines that we currently use. They are ready to provide 100 million doses for the US, and they have committed, I think, between 500 and 800 million for other areas of the world. And they are right now in trials for pregnant women. We have also heard a lot about the AstraZeneca. This one is approved in the EUA and is the one that is used in the majority of shots there. It's a 2-dose and it's like 82% effective. We also have heard about the Russian vaccine, Sputnik 5 from the Russian Ministry of Health. This is 2 doses and 92% effective. And the CanSino Bio, which is approved in China and is 1 dose with 66% effectiveness. I know that Sputnik and, or actually the Russian Ministry of Health and AstraZeneca are trying to work together to see if they can boost the effectiveness of the AstraZeneca vaccine. So there's a lot of development. So there's still a lot of work that needs to be done. But the more options we have, not only for us, but for the entire world, in the best shape we will be in when we achieve that. So how is the situation looking in the US? Well, I think probably we can say less than ideal, but we are getting there. There's progress. We are much better than we were a month ago. There is in California about 5.3 people have had 2 doses and 14% have had at least 1 dose. The majority of these vaccines have focused on vulnerable populations, including frontline workers, of course, healthcare workers, and people over 65. So right now, Santa Barbara County lowered down the threshold. It was 75 and above, and now it's 65 and above. And again, as the vaccines become available, we'll start vaccinating other groups, including educators and people in other industries. Another question that I get often is, what is the representation, right? How were these trials done, and what groups were they vaccinating in their trials? So what I can tell you is that they tried to recruit as many patients from diverse races and ethnicities to make it comparable to the population that we have in the US. We know that this disease has disproportionately affected our communities of color. So it would be ideal to have an overrepresentation of these groups in the vaccine trials. Unfortunately, the recruitment was not as effective as possible, but I can tell you that the diversity that we have, at least in the Pfizer and the Moderna, and I got information also from Johnson & Johnson, in terms of diversity and coverage of these different groups, is better than whatever we had before for other vaccines and also for other drugs. So there's at least evaluation across, as I said, races and ethnicities, that this vaccine is working the same way. So why should we get a vaccine? So we hear this term again all the time, herd immunity. What does that mean? Well, what that means is that at some point in the community, enough people are going to be immune to this disease, either by natural infection or by vaccination. And that is going to slow down the rate of transmission. So in the case that we have here, there is no herd immunity. Everybody is susceptible to this disease, which is the situation that we have had for the past months. We are all susceptible if exposed. So the transmission is going to be very rapid, which is what we have seen for the past year. However, when you start vaccinating people, or again, people start acquiring immunity naturally, the susceptible hosts that you have that can be infected by the virus are fewer, and that immediately mitigates the transmission and controls the disease. Unfortunately for this disease and for many others, if we wait for natural immunization, if we wait for people just to get it, many more individuals are going to die. And right now the toll, again, in the US is over 500,000, which is absolutely devastating. So we cannot afford to just wait for people to acquire natural immunity, and we have to be aggressive with these vaccination campaigns, trying to convince as many people as possible to get a vaccine. So how can I get a vaccine? I recommend all of you to visit all of these sites and register in every single one of them. So try, if you haven't tried to get a vaccine, you're protecting yourself and you're going to be protecting others, as I said, by just developing this herd immunity. So it's really an effort from all of us. So I know Cottage has a wait list, Samson has a wait list, the My Turn has the instructions on how to get registered and what happens once your turn comes up. There's immunizations in CVS, in Albertsons, in Ralph's. So there's a lot of avenues that we can have, especially we are 65 and above, to get a vaccine right now. And even if you're not, I still encourage you to register in the My Turn site, so that you get notified when your turn comes up and when you can get a vaccine. So in the last 5 minutes, I'm going to talk to you about the viral variants, which is something that is, of course, of concern to many of us, and we've seen the recent developments in the news. What I can tell you is that the development of these variants is not unexpected. Viruses are constantly changing, and especially those that have RNA as genetic material tend to be very error-prone. And what that means is when they replicate, they introduce little mistakes in their genetic sequence. And this leads to mutations. And those mutations accumulate and they result in genetic diversity. Some viruses are going to be more fit, which means that they can probably transmit better or survive better under some conditions. Some may cause different disease, as it's been suspected. Some may evade the immune response just because they look different enough from the original virus that you develop immunity to. That is going to just be not working so well for that particular virus. So there's a lot of changes that can occur that can have different consequences, but this is not unexpected. This is the general process, and this is the general behavior of viruses. Again, and as I said, some become better, some become worse. Some may become less infectious, and some may become less pathogenic, and some may cause less disease. So it can go in any direction. So what are these variants that we're talking about? Well, these are defined by a specific assortment of mutations. So we go and look at the viral genome and we start finding the changes. And then we start seeing that the more sequences we see, the more conserved those changes are. So we start seeing that the same things are kind of being transmitted across the population, and we can determine that as a variant that appeared. A new virus that is stable and is transmitting in the population with a specific set of mutations. So with all of this, what am I saying? What are these changes? What are the mutations? Well, this is, again, I love the sites from the New York Times. They are pretty good with their illustrations and with their explanations. So here what I'm showing you is some changes that have been detected, and there's many more, in the viral genome. So the genome is this squiggle right here, and now we're just looking at it as a representation here in this rod. And we're seeing that there is accumulation of these red lines in different places within the genome. One of the places that seems to have a lot of change, is not surprisingly, so I'm going to tell you in a moment, is the spike protein, right? So this protein that I tell you is kind of the key for the lock in the cell. And I told you that a lot of antibodies are probably made for this protein. It's one of the first things your immune system is going to see. Well, all of that immune response put this protein, or put this virus under pressure to evolve this protein and to change it, right? So the purpose of a virus is always going to be to replicate, to make more, to transmit. And if all of a sudden you find a hurdle, in this case an antibody that recognizes your spike, well, you change it. You just make a little tweak and see if all of a sudden you are not recognized by that antibody anymore. And we have a lot of viruses that are being made, and we have a lot of people that are making these viruses. So the chances to find a mutation that is going to be successful in evading that antibody, or that is going to be successful in being a better transmitter, or that is going to be successful in many other ways, just a virus that's going to be more fit, increases. The more people that carry this virus, the more the virus replicates, the more chances to accumulate mutations that are going to result in these changes that are going to promote the transmission. So it's not that the virus all of a sudden is changing more dramatically. It's not that the virus is acquiring a new capability of making the mutations. This is the normal process. It's just a game of numbers. We have a lot of people that are infected at the same time. And that, again, puts the scale in the favor of mutations that are going to be increasing viral fitness in some cases. So what are these strains that we hear so much about? So right now we have three strains of concern, or three variants of concern, that are these lineages, the B117, B1351, and P1. We may know them better from the place of origin, which are the United Kingdom, South Africa, and Brazil. It is concerning, or the main concern about these variants is that they seem to be more transmissible. And the way that we determine that is by looking at the sequences, at the genetic sequences of the viruses that are circulating in the community. And so people started seeing that all of a sudden, we kind of transition from having the same virus across the population to specific viruses in specific regions that all of a sudden took over. So we started detecting, let's say, some of these in September. And then we see that 20% of the population carry this virus in October. And then we see that 100% of the population carry this virus in December. So that tells you that this virus became the most prevalent, and it has a different pattern of transmissibility. It doesn't mean that it's going to transmit in a different way. It just means that the dynamics of transmission somehow are better for this particular strain. There's also the concern of increased severity. So you have to go and see, okay, what are these variants? How are these various variants correlating with the symptoms? Do we see all of a sudden that people are getting more sick? Do we see more hospitalizations? What kind of symptoms do we see? And of course, the potential of reduced vaccine effectiveness, which if, as I told you, if the virus changes a part that is recognized by the antibodies that you're making against it, all of a sudden it's not going to be recognized as well, and the effectiveness can drop. And that is something that is being monitored very carefully and very closely, not only for these strains, but other strains that are appearing. And that brings me to what probably you guys saw in the New York Times, in the LA Times, in all the times yesterday. So this idea that there is or this data that supports that there is a California strain that could be more infectious and more lethal. What I can tell you is that there's still a lot of studies that need to be done in this. We are seeing in the sequences that are being analyzed that there is this strain that is becoming more prevalent in the West Coast. However, just a couple of studies are coming out with the relationship between this strain and different symptoms or different outcomes of the disease and transmission. So I think we're just starting to learn what is going on with this California strain. We need to be alert, and we need to keep on looking to see what is circulating in our communities and how are these strains affecting, again, the outcome of disease. So yes, there are some studies. I think they're very preliminary. Some of these haven't even come out, so we cannot judge them. And there's still a lot of data that needs to come. So more studies and more information needs to be available before we make a definitive statement about this particular Cal-20 variant. But it's something that we should be aware and alert. So one of the most concerning points is the vaccines, right? So I can tell you that there is some different responses for these variants for the vaccines that are available out there, but so far those differences are not so drastic or so dramatic that they can make a vaccine completely useless in particular or for a particular strain. So yes, there might be some evasion of the immune responses elicited by these vaccines, but they still seem to offer a protection, especially for the severe cases, which again is one of our main, or should be one of our main objectives, trying to keep the pressure off our hospitals and prevent serious outcomes from this disease. That said, one of the most exciting advantages that the mRNA vaccines have is that they can be changed pretty quickly. So I can tell you that we can basically write little sequences of RNA in the lab. We can synthesize them very quickly, and we can assemble them, well, not we, but the companies can assemble them very quickly in these delivery packages. So they are very versatile. So the moment that we find a new strain, if there is the need for a booster, or if there is the need to develop a specific vaccine for a particular region of the world, these mRNA vaccines are pretty malleable, and they're pretty amenable for these kind of changes. So I know that Moderna reported, last month that they were developing a booster for one of these strains, which is the South Africa strain. So within a couple of weeks, I'm pretty sure that they can prepare this vaccine and start trying or doing some trials. And the FDA has also said that they are willing to allow a more streamlined process for the evaluation of these strain-specific vaccines, since they have already gone through the whole approval process and review process for these vaccines. So this is pretty encouraging. And even if we face some vaccines, what I want to bring your attention to is that people are thinking about this, scientists are thinking about this, not only for monitoring, but for prepare the response, and the companies are ready to deploy new vaccines if necessary. So what's the future? So I saw this quote from SNL the other day that made me think about a reality. So we see the light at the end of the tunnel, right? It's like we see it. But because we see the light now, we can see how stinky the tunnel is for real. So there is hope, but it's not immediate. The hope relies on our vaccination campaigns to be successful, on people to be compliant, on not letting our guard down, or basically maintaining a lot of the status quo that we have had so far that has helped us in prevention. We cannot start having gatherings and, you know, dropping again our guard right now. It is a critical moment that we are close to having this herd immunity. We're close to getting there, but it's not the end. We are close here in the US, but there are a lot of countries that still don't have access to vaccine, and because of globalization and movement of populations, there's always going to be the risk of reintroduction of the virus. We have these variants, and we have to be very careful. That could pose a risk of reinfection. But if we monitor them closely and we understand them, we're going to be able to control them. So there's a lot of things that still need to be understood better, and that we are getting very close, but we are not there yet. I know that Fauci said, you know, that we hear the end line moving. It's just a moving target constantly. So somebody said that the pandemic was going to be done by April. I hope they're right, but I'm not very hopeful that the pandemic will be over by April. Maybe the control here in the US will be better, but still, we're going to need to keep on using our masks, keep on washing our hands, keep on, you know, having these distancing and modified behaviors. And we're just going to have to face that there's going to be a new normal, and that things are going to start normalizing little by little. So I think Fauci has been a visionary, and it is possible that we will be wearing masks until 2022. I hope before that. With that, I want to finish. I went a little bit over time, but I'm happy to take questions. And last time I told you keep calm and wash your hands, and this time I'm going to tell you keep calm and keep on wearing a mask. Thank you.
Great, thank you so much. We really appreciated that. We have a bunch of questions in the chat, and so I guess maybe I'll just call on people to ask their questions. So John, you had a question about the second dose? Or did you want me to read them?
I see it. I have them here. So I can.
You have them there? Okay.
Yes, I can read them if you want. So the
Sure.
the second dose of the vaccine increases the effectiveness or the durability. So we have seen that it increases the effectiveness. So if you compare how good it is from the first shot to the second one, there's definitely a difference. So the first shot does offer protection, but the second one is certainly higher. So it does help. And that's why Johnson & Johnson is exploring the possibility of a second shot and seeing if it's gonna improve, you know, significantly the response. In terms of the durability, I think we still don't have enough information to see if it's going to extend the durability. And again, since having the two increased effectiveness, I think most people are recommending just go for the two. Because of availability, there are some areas that are going with just one, or exploring the possibility of just one, which still offers some protection. Okay. So does the protein approach dispel anti-vaxxer concerns since such vaccines contain no genetic material? Not really, unfortunately, because the anti-vaccine movement has a lot of tools under their belt, and they oppose a lot of things, not only the genetic material, but the adjuvants, the vehicles that you use, basically anything that is in a vaccine. There is in many cases non-science and non-fact founded argument for that. So no, unfortunately, it's still not helping. Let me see, we have a lot. Okay, so if your cells display the spike protein, will your immune system kill your cell? So some cells are just going to die and release the spike protein. Some cells may be targeted, but remember, this is a very transient response, right? It's not that you have the mRNA forever. This is pretty labile, and it's going to go away very quickly. It's going to be quick enough that you can generate a response. It's going to be quick enough that you are not going to have secondary effects from your cells being killed. It's not going to be damaging to your organism. Okay. Let's see. Okay, so I like this question of what do we do next? So the mRNAs are being so successful, right? what are we targeting? So I can tell you that they have programs for Zika virus, they have programs for HIV, they have programs for Ebola. There's a lot of things that we're targeting. I think HIV is one of our priorities as a society. It's like we could prevent it. You know, it's not a big deal in the US that much anymore. We still have a lot of cases, we can't treat them, but in other communities it's devastating. So I think HIV is a good one. And the great thing about the mRNA vaccines is that if we encounter a new virus, we're going to be able to respond rather quickly. Okay, let me see. Okay. Oh, good. So there was some confusion about the numbers. I think probably it was just, I gave percents. Is there any work so here is it. Is there any work to develop a self-spreading vaccine that's highly transmissible but not deadly? I have the feeling that because of all the concerns that a lot of people have about vaccines, something like that would not be very welcomed. And also these approaches that we have right now, yes, although they are dispendious in having to deliver and they are hard to reach populations, etc. It is still pretty important to know that they are done thinking about safety, right? If you have something that is spreading between humans that could offer help, but as I told you, viruses mutate, so the risk of mutations and the risk of acquisition of some other capacities, it's pretty serious. So I don't think that that's an avenue that anybody's going to explore. Okay, so if a vaccine is only effective as normal flu one, but COVID is 10 times more severe, does it mean that the J&J has 10x severe outcomes as the normal flu vaccine? No, so the Johnson & Johnson, fortunately, has a pretty good record of also preventing these severe cases. So we're seeing that even when the effectiveness, again, remember that the effectiveness is measured by displaying mild to moderate symptoms of COVID-19, not the severe ones. So in that regard, the efficacy may be a little bit better in preventing those mild symptoms, but it's pretty good preventing the severe symptoms and the severe outcome of the disease. So I just want to make very clear that the Johnson & Johnson is also pretty successful in that. And Bob wants me to talk about my CRISPR rapid test. So we, it was a great experience. So I told you probably last year that we were starting to thinking of developing a rapid test, which we did. We have one that is CRISPR based. We used it to do surveillance here in our community. So we did, we tested about 2,000 people, and we were able to find cases using our CRISPR test. And we also proved that this methodology is as sensitive and as useful to do screens in populations, especially asymptomatic populations, as the traditional RT-qPCR. So it works very well. We are navigating the complicated world of the FDA, which has been less than fun, especially for me as a researcher and not as a regulatory person. But what we're doing right now is trying to simplify it and probably adapt it to see if we can detect some of these viral variants, which we could with the CRISPR method. So it's evolving, and we hope to simplify it enough at some point that it can be delivered anywhere and that can be executed anywhere. So we have developed some other methods that allow us to handle samples, hopefully in the field. And the idea is to make it completely field deployable and again, versatile enough that it could help detect some variants of concern, especially in areas that need it. Okay.
Great, thank you. I think we have some other questions. I had a question, and that is there a virological reason why vaccines might produce a better immunity than getting the disease itself? And I saw that there were recommendations that people who had already had the disease maybe only get one dose of the vaccine.
Yeah, so there's a lot of interest and exploration of that, because one thing that we don't know is how long the immunity lasts, right? So if you got the disease, last year in June, we don't know if you're still going to be protected, now or 6 months from now. So it may be to kind of prolong a little bit that the natural immunity that you have. Does that answer your question?
Yep, thank you. Mary, you had a question?
Actually, there's a missed question in the chat, and that was asked by George Babor. What is the difference between mRNA and RNA? And since I don't know the answer, therefore,
Yes,
I mean, RNA is RNA.
yes. So messenger RNA is made by your cells hundreds of times daily. I mean, it is, again, the molecule that allows your genetic information to be decoded, right? So it is in your cells, it's there. Retroviruses are different. So retroviruses are going to carry their genome, right? And I understand where this question is coming from. So there is a lot of concern that this mRNA that we're giving as a vaccine is going to act as a retrovirus and is going to be incorporated in your genome as a retrovirus. And the short answer, without going into the virology class, is no. The retroviruses are going to have the capacity of accessing your DNA. They're going to have the capacity of going to the nucleus and getting there where your genetic material is. The mRNA that you have as a vaccine is not going to have that capacity. Okay. The second aspect is retroviruses are going to replicate, right? So they're going to generate more particles. They're going to increase the risk of infecting more cells. The mRNAs that you're delivered with the vaccine are going to go into the cells that they're going to go. They're not going to replicate, and they're going to be eliminated from the cells, right? So again, it's very important that we remember that this is a transient event. That is not something that that mRNA is not going to stay with you forever. That mRNA is going to be degraded, and then that's it. That was the last time you saw it. Okay.
So is there a particular code at the end of the mRNA that has it be destroyed immediately after? I mean, what is it that differentiates it?
So yes, the RNA has some modifications. So it doesn't look completely like one of yours, but again, it's more for stability that it has been changed, than rather for some other kind of modifications. So it has been modified, so it's actually a little bit more stable, but it's still going to be degraded ultimately. So it does look, it's a messenger RNA, it looks like yours with those modifications, but again, on purpose there for stability rather than anything else.
Okay.
Mm-hmm. Oh, Bob? Yes.
Yeah, Bob?
Thank you. Yeah, so in England, they are giving all the first doses before they're going to the second doses. Does that make sense from a public health standpoint?
That's a good question, and I'm not a public health expert, so I can give you my opinion. Don't quote me necessarily. I guess it depends a lot on the type of vaccine, right? So what level of protection do you see after the first dose compared to the full scheme of 2 doses, right? So for some, you may see more protection than for others. The availability of the vaccine, right? So there are some, it's not just in England, but there are some other communities that are exploring having 1 shot that versus a 2 shot while the supply arrives. What I can tell you is that the effectiveness or the efficacy of the vaccines with the 2 doses, we definitely see a change in the protection from the first dose to the second dose. And it is a good difference, right? So yes, it will offer protection, but it's not going to be the level of protection that you are offering with both doses. So that's why, we're maintaining the scheme. At least here in the US, is maintaining the scheme of the 2 doses and is asking people to be compliant with receiving the 2 doses. You are going to be having more protection.
And how do they determine how long they can wait? I mean, a lot of boosters, you get months or even a year later.
That is also a very good question, and there's a lot of information that is coming on the length that you wait between the first and second dose. So now AstraZeneca is saying, oh, maybe we wait longer, you actually have more protection, right? for me, it's not entirely clear of, what would be the difference between 1 or 4 weeks, or why choosing 2. I think it's basically what people have seen with the evolution of the immune response in some of these cases, and how it has been better when they do it at this point, right? Again, I think there's still a lot of research to be done in the length of the doses, right? On the space between the doses. So the problem is that some of the schemes that were already approved. So you cannot deviate much from those. So you have to go through new studies and see new data coming on how the responses are. Makes sense? Okay.
Skana, you had a question?
Yeah. So the Moderna has some extra coating or something that makes it able to exist in warmer temperatures. Does that coating also cause more reaction in people who are susceptible to reactions to vaccines and drugs and all kinds of other chemical things?
I don't have the numbers, but I think that they are very similar for anaphylactic shock reports between the Pfizer and Moderna. So, and again, it is rare, right? So 10 cases per million cases, or 10 cases per million people vaccinated is still pretty low. I think, I don't remember the numbers for peanut allergies, but are definitely a log or 2.
Sure.
So yes, it is of concern, and I know that in some cases, people that have reported anaphylactic shocks in the past, that's one of the questions they ask you, right? When you're going to get a vaccine, so they ask you, have you had this before? And if so, they may recommend you talking to your doctor to see the risk-benefit conditions. So I don't think it's necessarily just the coat in the or anything particular about the coating in the Moderna. Is just the delivery, right? And again, as I said, I don't see much difference in the numbers, so I don't recall.
Okay, thank you.
Yeah.
Okay, any more questions from anyone?
I have one more question. It's kind of silly, but how many molecules of messenger RNA are in each dose?
Oh, that's a great question. I don't know. I don't know that. I will look it up. They may not even tell you. Some of these things are highly proprietary, so they may not go and reveal all their secrets, but I remember reading it somewhere. I don't recall. I don't know. You got me on that one. Okay.
Anyone else? Okay, well, thank you very much.
Thank you.
Really appreciate your talk, and we enjoyed it so much. So
Thank you. Good to see you again.
Good to see you. Take care.
Thank you for doing this.